Every single year, millions of people pay surgeons, a small fortune, to vacuum fat out of their bodies, only to just throw it immediately in the trash.
Right. Goes straight to biohazard waste.
Exactly. I mean, we freeze it, we burn it, we treat it like this inert, stubborn enemy that we're just constantly at war with. But what if the very tissue you're trying to sweat off on the treadmill is actually a pharmacy?
Yeah, that's the paradigm shift we're looking at today.
Right. What if people are throwing away literally the most advanced, customized regenerative medicine on the planet? Welcome to the deep dive. Today, we're exploring the hidden superpowers locked inside human fat tissue, stem cells, and these microscopic messengers called exosomes.
And it really requires a complete shift in how you view your own body.
Yeah.
I mean, we're looking at emerging therapies that go far beyond just managing the symptoms of an injury or, an illness.
Right, not just putting a Band-Aid on it.
Exactly.
Yeah.
We're talking about actual physical tissue regeneration and repairing severed neurological pathways.
And for you listening, to really get to the bottom of how this works, we've pulled together a fascinating stack of medical literature.
It's a really diverse stack, too.
Oh, absolutely. We are synthesizing everything from clinical surgical notes on treating severe Crohn's disease to breakthrough laboratory research on diabetic and chemotherapy-induced nerve pain, all the way to the practical, real-world application of something called nanofat from Tulip Medical.
Yeah, the real-world applications are just wild.
They really are. So the mission for this deep dive is to figure out how the body's own adipose tissue fat basically can be processed and used to literally reprogram damaged nerves, halt chronic pain, and heal tissues that just stubbornly refuse to heal on their own. Okay, let's unpack this.
Let's do it.
To understand these cutting-edge microscopic mechanisms, we first have to look at the physical raw material doctors are actually using in clinics today.
Which is, simply put, the fat itself. But historically, when doctors talk about fat grafting, they're usually talking about volume.
Right, like cosmetic fillers.
Exactly. Taking fat from the abdomen or the thighs and moving it somewhere else to act as a natural filler. That's essentially what the Tulip medical documentation refers to as microfat.
Microfat, okay.
Right, so microfat maintains the cellular structure of native fat, and surgeons typically inject it under the skin using needles or cannulas that range from like 18 to 25 gauge.
So small needles, but definitely not microscopic.
Yes, exactly. But the literature quickly pivots to something entirely different called nanofat. And with nanofat, the harvested fat is heavily processed and sized down to what is called the tissue stromal vascular fraction, or TSVF.
Hold on. Tissue stromal vascular fraction, that is a heavy piece of medical jargon right there. What does that actually mean if you're like holding a syringe of it?
Right, let's break that down. Think of normal fat like a bunch of water balloons.
Okay, water balloons.
The balloon itself is the cell membrane, and the water inside is the lipid, the actual oily fat that stores your calories. To get to the stromal vascular fraction, you're basically popping all those balloons, washing away all the oil, and keeping only the rubber casing.
Wow, okay, so just the structure.
Just the casing, the complex network of blood vessel fragments, collagen, and most importantly, the stem cells that were hiding between those balloons.
Oh, I see. So you're stripping away the actual fat part of the fat.
Exactly, the lipid is gone.
Which totally explains why the documentation notes that nanofat has absolutely zero volumizing properties. Like if a doctor injects it, it won't puff up your skin or add any structure at all.
None. It's purely regenerative. And because it's been emulsified down to this cellular matrix, it can be injected with tiny hair-thin 30-gauge needles.
Which allows for incredibly precise application in very delicate areas, right?
Precisely. For instance, the Tulip Gold D kit uses a closed syringe system to safely harvest this tissue. It's a low-pressure, gentle acquisition that keeps the biological material alive and viable.
Because you don't want to kill the stem cells while you're extracting them.
Right, that would defeat the whole purpose. And the clinical applications being tested right now are staggering. Urologists, like Dr. Elisa Bergandini mentioned in the sources, are using this for urethrovaginal fistulae, erectile dysfunction, urinary incontinence, and severe perineal scarring.
Yeah, and there was one case study in those clinical notes that absolutely blew my mind. It involved a patient with a complex anorectal fistula caused by Crohn's disease.
Oh, yeah. That is a notoriously difficult, just devastating condition to treat.
Right. And for those who don't know, a fistula is an abnormal tunnel that the body creates, connecting two organs or connecting an organ to the skin.
And in a patient with Crohn's disease, the constant severe immune response and inflammation make these tunnels incredibly resistant to closing up and healing. They just stay open.
Yeah, and this specific patient had failed every standard treatment. I mean, the doctors had surgically placed a aceton drain to keep the tunnel from abscessing. The patient was on heavy biologic drugs, specifically infliximab, just to suppress the immune system, and the tissue still simply refused to heal.
It's a desperate situation for a patient to be in.
Truly, so the surgeons lipoaspirated a small amount of fat from the patient's abdomen, processed it down into just 40 milliliters of this nanofat, and injected it directly into the walls of the fistula tract.
And the results were incredible.
6 months later, the tunnel was completely healed. No recurrence.
It is a remarkable outcome for a condition that normally leads to repeated, highly invasive surgeries and a drastically reduced quality of life.
But wait, hold on. This is where my brain just gets completely stuck. Making nanofat sounds like, I don't know, putting a brick wall into a blender to extract the liquid blueprint of the building.
That's one way to put it.
Right. So how does taking blended belly fat and injecting it into a severely inflamed, infected fistula tunnel not just make the infection worse? I mean, how does fat actually heal a chronic inflammatory wound?
Well, that is the core mystery here, and answering it takes us from the macroscopic level of the fat down to the microscopic level.
Okay, zooming in.
Exactly. The fat cells themselves, the adipocytes, they aren't doing any of the heavy lifting. As we discussed with the stromal vascular fraction, the oil is gone.
They're just the packaging that gets discarded.
Right. The real functional drivers are the mesenchymal stem cells, or MSCs, that live within that adipose tissue. Human fat is incredibly rich in these stem cells.
More than bone marrow, right?
Far more, yes. But even then, the mechanism isn't what we thought it was for the last 20 years. It's not just the stem cells themselves doing the healing. It's what those MSCs are secreting into the environment around them.
Ah, okay. You're talking about the exosomes.
Yes. What's fascinating here is that for a long time, the medical community believed stem cells healed tissues by migrating to the site of an injury and grafting themselves into the damaged area and physically transforming into the new tissue.
Like, if you needed heart muscle, the stem cell literally became heart muscle.
Exactly. That was the working theory. But recent science shows that's not the primary mechanism at all. They actually heal through paracrine signaling.
Paracrine signaling, let's break that down for the listener. That essentially means cellular texting, right? Chemical communication between neighboring cells.
That's a perfect analogy. It is highly localized communication. And the physical medium of that communication is the exosome. Exosomes are endosomal membranous nanovesicles.
And when we say nano, we mean almost unimaginably tiny. I mean, the sources clock them at 30 to 200 nanometers in diameter.
Which is hard to even conceptualize.
Right. For context, if a standard human cell was the size of a massive sports stadium, an exosome would be roughly the size of a football.
Wow. Yeah, they are microscopic communication envelopes. When a stem cell recognizes that the tissue around it is damaged or inflamed, it packs these little lipid envelopes with a highly specific cargo.
What kind of cargo are we talking about?
We are talking about genomic materials, specifically microRNAs, as well as various proteins and lipids. The stem cell then ships these envelopes out to the damaged cells.
So if you or someone you know suffers from chronic nerve pain, these tiny envelopes are essentially carrying the exact stop hurting and start repairing instructions that those damaged, glitchy nerves desperately need to read.
Yes, they carry the exact instructions needed for repair.
You know, I like to think of exosomes as tiny biological USB flash drives. Like, the damaged cell is a computer that is glitching out running a toxic inflammatory program on a loop.
I love that.
Right. And the stem cell plugs this little exosome USB drive into the damaged cell, uploads a vital software update, and just fixes the broken code.
And not only do they upload the software update, but the exosome cargo perfectly mimics the healing and immunomodulatory properties of the parent stem cell that created it.
But that brings up a massive question for me. If these tiny USB drives are the things actually doing all the work, if they are the ones delivering the software update, why did the medical field spend decades obsessing over transplanting the whole stem cell?
It's a great question.
Why were we injecting the whole computer when we just needed the flash drive?
Basically because the technology to isolate, purify, and even understand exosomes simply wasn't there until very recently. We observed that stem cell therapy worked, but we completely misunderstood the mechanism.
We were looking at the wrong part of the process.
Exactly. And honestly, moving away from whole stem cells and focusing strictly on the exos solves a lot of massive problems in regenerative medicine. Whole stem cells come with a lot of clinical baggage.
What kind of baggage? Like, what are the risks?
Well, when you transplant live whole stem cells, you run the risk of immune rejection if those cells aren't autologous, meaning harvested from the patient's own body.
Okay, that makes sense. Your body attacks foreign cells.
Right. But even if they are your own cells, whole cells are physically large on a microscopic scale. If they're injected into the bloodstream, they can get trapped in the microvasculature like the tiny capillary beds in your lungs.
Oh, wow.
And there's always the underlying risk of malignant transformation.
Wait, malignant transformation. Are you saying stem cell therapy can cause cancer?
It's a known risk, yes. Because stem cells are living entities designed to multiply, if they are placed in a volatile environment and receive the wrong chemical signals, they can proliferate uncontrollably and form tumors.
That is terrifying.
It is.
Yeah.
But exosomes completely sidestep that danger. They are not alive, they do not have nucleus, and they absolutely cannot multiply. They're just the messenger payload.
So they deliver the message and then dissolve, basically.
Yes. This gives them an unparalleled, targeted ability to hack the local immune system without the risks associated with whole cells. And they do this primarily by reprogramming a specific type of white blood cell called a macrophage.
Okay, so if these exosomes are delivering a software update to the immune system, what is the glitch they are trying to fix in the first place? Let's talk about why chronic nerve pain is so incredibly hard to stop.
It all comes down to a process called Wallerian degeneration.
Wallerian degeneration.
Right. When a peripheral nerve is injured, say it's crushed by a herniated disc or severed in a car accident, the part of the nerve fiber that is disconnected from the main cell body starts to die and physically break down.
It's cut off from its power source.
Exactly. And the Schwann cells, which act as the protective insulation around the nerve, they start panicking. They release distress signals that call in the immune system to clean up the dead cellular debris.
And the first responders to that distress signal are those macrophages you mentioned?
Yes. Specifically, they arrive in a state we call the M1 phenotype. M1 macrophages are highly aggressive and pro-inflammatory. Their job is to release toxic cytokines like TNF-alpha and interleukin-1 beta.
Which sounds bad, but it has a purpose, right?
And it does. They are there to destroy potential pathogens and clear out the dead tissue so an infection doesn't take hold.
Okay, so the M1 macrophages are like firefighters showing up to a burning house. They are aggressively chopping down doors and smashing windows with axes to stop the fire and clear the wreckage.
That is a really great way to picture it. And in a healthy healing process, once the debris is cleared, those macrophages are supposed to receive a signal to switch their behavior.
They need to calm down.
Right. They transition into the M2 phenotype. M2 macrophages are anti-inflammatory. They stop releasing toxins and instead release interleukin-10 and neurotrophic factors that actively promote tissue repair.
So the M2s are the carpenters who arrive to rebuild the house after the firefighters leave.
Precisely. But in chronic neuropathic pain, the system gets stuck. The transition never happens.
Oh, no.
The macrophages stay locked in that hyperinflammatory M1 state. The firefighters just keep swinging their axes, attacking the surviving nerve tissue, creating a continuous toxic loop of inflammation and pain signaling that the body just cannot break on its own.
And this is where the stem cell exosomes come to the rescue. The literature outlines a fascinating laboratory study on rats with an L56 spinal nerve ligation.
Right, the nerve ligation model.
Yeah, which is basically a surgical model used to simulate severe chronic neuropathic pain. They gave these rats an intravesical injection, meaning an injection right into the spinal fluid of exosomes derived from umbilical cord stem cells.
And the researchers were looking to see if the exosomes could force what we call macrophage polarization.
Meaning forcing them to switch from M1 to M2.
Exactly. And the results were profound. The exosomes physically entered the aggressive M1 macrophages, delivered their microRNA payload, and successfully reprogrammed them to become M2 macrophages.
Wow. So the exosome is essentially the project manager showing up on site, telling the axemen to go home and handing out hammers to the carpenters.
That's exactly what happens. And we can measure this shift at the molecular level. The study showed that after the exosome injection, the toxic markers in the spinal fluid, the TNF-alpha and IL-1 beta plummeted.
The fire was put out?
Right. And meanwhile, the healing markers, like IL-10 and brain-derived neurotrophic factor, or BDNF, surged.
And how did that actually affect the rats in the real world, like behaviorally?
Their mechanical and thermal hypersensitivity was completely reversed. In chronic nerve pain, a light touch or a mild temperature change can feel agonizing.
Right, like wearing a shirt is painful.
Yes. The exosomes completely eliminated that hypersensitivity.
If we connect this to the bigger picture, We
Hmm.
realize this isn't just masking pain. When a patient takes a heavy opioid or a traditional nerve medication like gabapentin, they're really just turning down the volume on the fire alarm in their brain.
While the fire in the tissue is still burning.
Exactly. But by forcing macrophage polarization, these exosomes are putting out the fire and repairing the alarm system itself. They are changing the cellular environment to permanently turn off the pain loop.
It is actual fundamental regeneration, not just palliation.
But wait, if they fire the M1 macrophages and fix the inflammation, what about the actual broken nerve fiber? How do they fix the severed wire?
That is the second and arguably even more incredible function of the exosomes. Once the inflammation is cleared, they physically command the severed nerve fibers to regrow using the genetic code packed inside them.
That is wild.
And this is where we have to look closely at the specific microRNAs, or miRNAs, that make up the exosome payload.
You mentioned those earlier. What is a microRNA actually doing to a nerve cell?
So miRNAs are small, non-coding RNA molecules that regulate gene expression. They don't build proteins themselves. Instead, they bind to messenger RNA and can literally turn certain cellular processes on or off.
They're the switches.
They are the switches, and the sources highlight a few specific switches that are absolute game changers for notoriously difficult conditions like diabetic peripheral neuropathy, or DPN, and chemotherapy-induced peripheral neuropathy, CAPN.
And those are conditions where patients lose feeling in their hands and feet or experience this constant burning sensation, and traditionally doctors just shrug and say, the nerves are dead, we can't fix it.
But the exosomes carry specific tools to intervene. For instance, the literature points to miR-146A. When this microRNA enters the tissue, it aggressively reduces inflammation and actually restores the physical integrity of the blood-brain barrier.
Oh, wow.
Then you have miR-133B. This microRNA directly regulates the length and the number of growing nerve fibers. It binds to the neuron and sends a continuous signal saying, keep growing, branch out, extend.
So it is actively hitting the gas pedal on nerve growth.
Exactly. And perhaps most importantly, there is miR-181C5P. This specific microRNA targets and inhibits a cell death mediator known as BIM.
Okay, how does a microscopic piece of RNA stop a cell from dying?
Think of BIM as a self-destruct button inside the neuron. When a nerve is severely damaged, BIM is activated to safely kill the cell.
Apoptosis.
Right. The miR-181C5P physically blocks that self-destruct button from being pressed. By inhibiting BIM, the exosome effectively hits the biological override switch on cell death, keeping the injured neuron alive long enough for the other microRNAs to repair it.
Here's where it gets really interesting. There was a study mentioned in the literature on mice with ovarian cancer. They administered exosomes derived from cerebral endothelial cells alongside a platinum-based chemotherapy drug.
Right.
And normally, that chemo drug is brutal on the nervous system. It causes severe toxic nerve damage.
That is the chemotherapy-induced peripheral neuropathy we mentioned earlier. It's a major limiting factor in how much chemo a patient can actually tolerate.
Right. But in the study, the exosomes not only reduced the toxic nerve damage usually caused by the chemo, they actually increased the anti-tumor effect of the drug. How is that even possible? I mean, how does it fight the cancer while building a shield around the nervous system?
It happens because the exosomes are altering the tumor microenvironment. While they're sending survival signals to the healthy neurons, they are simultaneously making the cancer cells more susceptible to the platinum drug.
That is unbelievable.
It's the absolute holy grail of oncology support, really. Protecting the healthy tissue while actively enhancing the cytotoxicity against the tumor.
But hold on, I have another skeptical question here. If you inject these exosomes into the bloodstream or even into the spinal fluid like in the rat study, how do they find the severed nerve?
Ah, the targeting question.
Yeah. Do they just float around blindly in the blood, hoping to accidentally bump into a damaged nerve in a toe?
It's a vital question, and it highlights the sheer sophistication of these nanovesicles. They don't just float blindly. Exosomes possess remarkable homing abilities.
Homing abilities, like a pigeon.
Kind of. The surfaces of these tiny lipid envelopes are studded with specific proteins and receptors that constantly interact with the environment. When human tissue is damaged, it doesn't just sit there quietly. It releases chemical distress signals, chemokines, and cytokines.
Like firing a biological flare gun into the bloodstream.
Exactly. The exosomes detect these chemical gradients. They sense where the concentration of distress signals is highest, and they physically migrate toward the source.
That is fascinating.
In several studies, researchers fluorescently labeled these exosomes so they could track them under advanced imaging. After a systemic injection into the bloodstream, the exosomes didn't disperse randomly at all.
They went straight to the injury.
They navigated exactly to the site of injury. In the nerve damage models, they accumulated heavily in the L5 spinal dorsal horn, the dorsal root ganglia, and within the actual severed peripheral axons.
They're essentially microscopic smart bombs, but instead of explosives, they are delivering medical supplies exactly to the GPS coordinates of the injury.
And because of this precise homing ability, combined with their incredible safety profile and their targeted genetic payload delivery, the entire regenerative medicine community is now moving rapidly toward what we call cell-free therapy.
Cell-free therapy, meaning we just leave the actual stem cell in the lab.
Yes. We discussed the baggage of whole stem cells earlier. The immune rejection, the trapping in the lungs, the tumor risks. Exosomes bypass every single one of those risks. They have very low immunogenicity.
Because they don't have the surface antigens that trigger an immune response, like a whole foreign cell would.
Furthermore, they are highly stable. You can isolate them, freeze them, and transport them across the world without having to keep complex living cells alive in a nutrient broth. They are incredibly safe and versatile.
It's like mailing the crucial letter to the damaged nerve without having to physically mail the postman along with it.
Exactly. And the delivery systems being tested right now are pushing the absolute boundaries of biomaterials. The sources mentioned loading these exosomes into polydopamine-modified chitin conduits.
Okay, stop right there. What on earth is a chitin conduit?
Chitin is a natural, tough, structural material. It's actually what makes up the exoskeletons of crabs and insects.
Okay, crab shells, got it.
Scientists are engineering biodegradable tubes out of this material. In cases of severe trauma where a nerve is completely severed with a physical gap between the two ends, surgeons stitch the severed ends into this tube.
Oh, I see. A literal bridge.
Right. The tube is packed with exosomes, providing a sustained, slow release of those regenerative microRNAs as the nerve slowly grows through the conduit and reconnects.
That is science fiction level surgery.
They're also testing delivery via intranasal sprays to repair complete spinal cord injuries.
Nasal sprays for a spinal cord injury.
Yes, because exosomes are so unimaginably small, they can cross the blood-brain barrier via the olfactory pathways in the nose, traveling directly into the central nervous system without needing a spinal tap.
This raises an important question. If this is so miraculous and it avoids all the risks of traditional stem cells, why isn't it available at every local clinic right now?
The bottleneck right now is manufacturing and standardization. How do we scale up, mass produce, and standardize these nanovesicles so they have a consistent, predictable payload every single time?
Right, quality control.
If a doctor is prescribing an exosome treatment, we need to ensure that a vial in New York has the exact same therapeutic microRNA concentration as a vial in Tokyo. Isolating, purifying, and characterizing biological nanoparticles at an industrial scale is technically daunting,
I can imagine.
but the investment in this space is massive, and we're getting there much faster than anyone predicted.
So what does this all mean for you, the listener? Let's take a step back and look at the incredible journey we've just taken. We started in a local surgical clinic looking at how a specialized closed syringe system extracts common body fat. We saw how that fat, processed down into the structural matrix of nanofat, could heal a severe, supposedly unhealable Crohn's fistula when nothing else could.
We then zoomed all the way down to the 30-nanometer world of the exosomes hiding within the stem cells of that fat.
We watched those tiny USB drives plug into hyperactive M1 macrophages, reprogramming the immune system's firefighters to put down their axes and start rebuilding the tissue. And finally, we saw how specific microRNAs can physically block the self-destruct button on dying cells and command severed nerves to regrow.
It completely reframes our relationship with our own biology. It takes something we view as a burden and reveals it as a profound medical resource.
It really does. It means that the very body fat society often tries to get rid of or shames people for having actually contains the ultimate customized regenerative medicine. The future of treating untreatable chronic pain might literally already be inside you.
The pharmacy is built in. We just finally found the molecular key to unlock it.
It leaves you with a pretty wild thought to chew on as we wrap up. If we can engineer these cell-free exosomes to deliver specific marinase that halt pain, regrow severed nerves, and actually enhance chemotherapy, could we eventually program our own custom exosomes to constantly repair our nerves as we age?
Oh, that's a fascinating idea.
Right. Could we effectively cure the physical decline of the nervous system before we even feel the first ache of growing older? Imagine a world where a quick nasal spray of your own engineered exosomes keeps your nervous system perfectly intact and pain-free at 90 years old.
It would change the entire biological definition of aging.
It absolutely would. Thank you so much for joining us on this deep dive. Keep asking questions, keep looking closer, and we'll catch you next time.